In this video, we're looking at three evolution topics, 7.7 through 7.9, common ancestry, continuing evolution, and phylogeny. The idea behind common ancestry is a relatively simple and straightforward one. It's the idea that groups of organisms share a now extinct ancestor. Of course, the farther back in time we go, common ancestors to larger and larger groups of still living species would be found. About 5 million years ago, the common ancestor for humans and chimpanzees existed. Going back a couple million more was the common ancestor to humans, chimps, and gorillas. A bit farther back was the common ancestor to all primates. Nearly 4 billion years ago, we would find the last universal common ancestor for all life on Earth. The evidence that we use to support common ancestry is based on cellular and molecular evidence, and it gives us clues as to the links between currently existing species and their ancestors of the past. Phylogeny is the study of the evolutionary history of organisms and groups of organisms, as well as the relationships between them. It involves the study of characteristics that are common to multiple groups of organisms and those characteristics that are unique. Probably some of the most familiar models being used in biology, specifically evolutionary biology, phylogenetic trees and cladograms are both used to represent those relationships. One of the most frustratingly widespread misunderstandings about evolution has to do with the relationship between humans and other organisms. It is the incorrect idea that humans evolved from or descended from apes. Images like these help to propagate that fallacy. It suggests that some chimpanzee-like organism changed a little bit to yield some primitive humanoid organism. And with enough change accumulated, there's us at the far right. This is wrong. What evolution and its evidence tells us is that organisms share a now extinct common ancestor with one another. Similar species have common ancestors that lived in relatively recent past, whereas dissimilar species had common ancestors that existed much farther back in time. It is the forces of natural selection that amplified the differences between groups of individuals within a species. As differences accumulate over many generations, eventually, the descendants of those groups of organisms are so different, they're no longer the same species. A video later in this unit will explore that process of speciation. The first piece of evidence we're going to look at is based around membranes. The fact that species possess membrane-bound organelles provides evidence of common ancestry. One of the most interesting pieces of evidence surrounds the serial endosymbiosis theory. If you don't recall the basic tenets of this scientific theory and the evidence that support it, be sure to review the presentation on topics 2.10 and 2.11. This theory explains to us not only the presence of chloroplasts and mitochondria in eukaryotic organisms, but also the entire endomembrane system, which includes structures such as the endoplasmic reticulum and the Golgi apparatus. The next category of evidence that supports common ancestry involves comparing chromosomes themselves. Of course, we can study the sequence of nucleotides between different species, but we can also explore the relatedness of species by comparing the genes themselves, as well as entire chromosomes. The presence of conserved genes that are found across a variety of species can help us to identify how related or not two different species are. For example, if we were to compare the rate of gene conservation in humans and chimpanzees, we would find a large proportion of our genes are perfectly the same as those of a chimp. Comparing our genes to more distantly related organisms, like mice, chickens, and fruit flies, we find that the percentage of those conserved genes goes down. What this indicates to us is that the common ancestor that we share with chimps existed more recently in time than the common ancestor we share with those other groups of organisms. We can also look at the similarities of entire chromosomes as a piece of evidence that supports the idea of common ancestry. One of the most fascinating pieces of evidence that points to common ancestry between humans and chimps can be observed by looking at our chromosomes. We know that humans have a total of 46 chromosomes, whereas chimpanzees have a total of 48. There's something peculiar about human chromosome number 2, which is one of our largest. As expected, at both of the linear ends of our chromosome, we find telomere sequences exactly where we'd expect to find them. At the center, joining the P and Q arms of the chromosome together, we find centromere sequences, just like we would expect to find. But strangely, we also observe another sequence that is just like a centromere in the lower Q arm of the chromosome. Also, there are telomere-like sequences right smack in the middle of the chromosome. Obviously, that's not what we would expect, unless we consider common ancestry with chimpanzees and observe chimpanzee chromosomes number 12 and 13. Individually, those chromosomes are arranged in exactly the way we would expect, with telomere sequences at the ends and a centromere sequence linking the two arms together. But the fact that chimps have 48 chromosomes and we only 46 suggests to us that those two chromosomes, at some point in the past, just after diverging from our common ancestor, would have fused together in the lineage that leads to us. The last piece of evidence for common ancestry involves introns. When we think about introns, at least in a very basic way, we think of them as not really serving a purpose in the expression of our genes. They don't hold coding information that will be used in the translation process. After all, they are removed during RNA processing, leaving only the exons behind. The connection between introns and common ancestry in evolution is that evidence exists that the presence of introns actually serves an evolutionary importance. Introns are believed to exist as sites for the recombination of genetic material, producing new exons. The evidence that we can use to support this idea is the phenomenon of alternative RNA splicing. This model illustrates a segment of DNA being transcribed into RNA and then ultimately being translated into a polypeptide sequence. But which exons are expressed is going to result in a different sequence of amino acids and therefore a differently shaped protein. The exact same gene in DNA can be used to produce not just a single protein, but multiple proteins by treating exons as introns. You may recall from an earlier video that evolution has neither a goal nor an endpoint. The evolution of species is an ongoing, never-ending process. Because environmental conditions, which include both living and non-living factors, are always changing, that means that natural selection forces don't apply consistent or even the same type of selection. In a given species, the phenotypic variations that are selected for at this moment in time aren't necessarily the same variations that were selected for in the past. By extension, the variations selected for by natural selection forces in the future may also be different. Generally speaking, trying to observe evolution actually happening is difficult. In some cases, it may be due to the fact that some organisms simply don't leave enough offspring for us to observe and analyze. Also, The generational time of some species is simply too long for us to observe enough generations and see change within the species. But with strong enough natural selection forces, and if species produced vast quantities of offspring and do so in a relatively short period of time, we can actually observe the results of evolution. In agriculture, we use a variety of pesticides to eliminate organisms like weeds and insects that are harmful to our crops. The pesticides are quite successful in eliminating those that have low resistance to it. But, thanks to the variations between individual organisms, some have a naturally greater resistance to the pesticide. Those are the individuals that survive and create offspring that also have greater resistance. This same kind of phenomenon is also observed in the evolution of bacteria. The use and often overuse of antibiotics has selected for bacteria with greater resistance to them. It is our own overprescription of antibiotics and inclusion of them in daily use products like hand soap that has driven bacteria to become more resistant. Even viruses in the gray area between life and non-life can evolve. Inside a host cell, they replicate and occasionally make mistakes when copying their genetic material. To the benefit of the virus and detriment to the host, an occasional mutation might result in a more virulent or more deadly version of the virus. It is the host, and specifically its immune system, that is providing the natural selection pressure on the virus. Take a look at the video linked above to learn more about the evolution of the coronavirus, SARS-CoV-2. Phylogenetic trees and cladograms are both models used to show the relationships between and among different groups of organisms. In this very small, simple example, we can see that we're looking at the order Carnivora, so these are carnivorous animals. There are 5 sample organisms from that order. From this diagram, we understand the relationship between leopards, a type of big cat, as well as canines like coyotes and the gray wolf. Models like these are not only used to show extant, or still living species, but they can also be used to show extinct ones as well. This is really useful in allowing us to study the relationships between currently living species and the ones that are no longer here. In this model, we can see down at the bottom Homo sapiens, that's us, and the relationship between us and 2 close relatives of ours, the common chimpanzee and the pygmy chimpanzee, or bonobo. There are a number of examples of extinct hominids as well, including some others that belong to the same genus as us: Homo habilis, the tool user, Homo erectus, the one that stands upright, and Heidelbergensis and the Neanderthals. This model is also useful because of the way it is illustrated. It shows some characteristics that are present in these different groups of organisms. We're going to take a look at how those are studied later. One of the differences between a phylogenetic tree and a cladogram is that phylogenetic trees are typically used to show how much time, geologic time, or sometimes how much genetic change has accumulated from one organism to another. So if we look at these primate trees again, we can see that some of the branches are longer than others. That is not accidental. Let's use this one as an example. The length of the line is used to represent that more time has passed between this point and this point, as opposed to between these 2 points. So when analyzing a phylogenetic tree, it's important to remember that the organisms that we see on the right-hand side are more recent examples, and then as we move left, we're going back in time and finding more distant connections and ancestors. There's more to a phylogenetic tree than time and relationships. It's actually quite simple because the way they're constructed is in a very logical, systematic way. The first thing that we need to recognize is that the clades, or branches, are used to represent a species or even a group of species. Here are a couple of examples. Each of the branches in the tree on the left represents an individual species, like the leopard or European otter. In the example on the right, it shows the relationship between land plants, but rather than individual species being represented by the clades, entire groups of organisms are clustered together. All angiosperms, or flowering plants, are included within this category right here. All ferns are included within this category, as well as some fern relatives. Another characteristic of a phylogenetic tree are the points at which those branches meet one another. Those points are called nodes. The nodes are significant because they represent the common ancestor for all of the branches that protrude from it. So, for example, in the left phylogenetic tree, this node would represent a common ancestor for the American badger, the European otter, the coyote, and the gray wolf, because all of the branches that extend or protrude from this point belong to those groups. In this right tree, this node represents the common ancestor to all forms of land plants. One of the things that you should recognize at this point is the farther back in time we go, the common ancestor is going to include more extant and, of course, extinct species. They can also be used to show the presence of shared characteristics. In this phylogenetic tree from earlier, each of the pink marks along the way are used to illustrate characteristics that are present in all of the species after the mark is indicated. This mark, for example, smaller jaws and cheek teeth, long legs, arched feet, well suited for long distance walking and running, and a larger brain, tells us that every species after it also has that characteristic. That characteristic would be true for Homo erectus, Homo neanderthalensis, Homo heidelbergensis, and us, Homo sapiens. The study of how scientists classify and group organisms together is called cladistics. It's the method that we use to group organisms based on their connection to common ancestors. In this diagram, we have the exact same phylogenetic trees, but as you can see, in each sample, a different group of organisms is being highlighted. This first group highlighted is referred to as a monophyletic group. The reason why is because it includes a single common ancestor, indicated by the blue circle with the number 1 in it, and all of the descendants from that common ancestor. In the second example, called a paraphyletic group, we can see a common ancestor, number 2, being identified, and some, but not all, of the descendants from that common ancestor. Because group G was excluded, we refer to this as a paraphyletic group. Had G been included, so that D through G were all highlighted, then we would refer to it as a monophyletic group. In the final example, a polyphyletic group, we can see that there are 2 common ancestors identified, but not all organisms that are descended from them are included. A polyphyletic group and paraphyletic group are really similar to each other in that not all of the descendants are included. But what makes polyphyletic different from paraphyletic is that it includes not 1, but 2 common ancestors. How are phylogenetic trees constructed? We use a variety of evidence to do so. Before genetic evidence became available, morphological evidence, such as the presence of certain characteristics or features, was commonly used. Derived and shared characters are used to help construct a phylogenetic tree. A shared character is one that is present in multiple groups of organisms. For example, we could say that we have a shared characteristic with leopards because we're both mammals. A derived character, on the other hand, is something that evolved specifically in that lineage leading up to that group of organisms. It's essentially something that would be special or unique to just that clade. In this example, we start off by collecting data on particular characters. On the left is what's called a character table, which identifies what kinds of features are present in different groups of organisms. The organisms that are included are a lancelet, a lamprey, a bass, a frog, a turtle, and a leopard. The characters used to create this particular tree are whether or not a backbone is present, whether or not the organism has hinged jaws, if it's got 4 walking legs, if it has an amnion, which is a membrane that surrounds a developing embryo, or if it's got hair. Every place indicated with a 0 represents that character is absent in the species. The 1s, on the other hand, tell you the character is present in the species. Let's start off with an easy one. The leopard on the far right. A leopard has a backbone, it has a hinged jaw, 4 walking legs, an amnion, and has hair. All of the characters in the table. On the other side, we see what's called an outgroup. We call this an outgroup for a couple reasons. 1, because it is least like everything else in the table, and 2, because it doesn't have any of the characters being tracked. The lancelet is the outgroup in this particular example, unique as compared to all of the rest of the species because it possesses no shared characters. The phylogenetic tree is constructed such that organisms that have the most shared characters are on branches closest to one another. Since the turtle and the leopard have the most shared characters, they are placed on adjacent branches. Next in line would be the frog, since it has 3 shared characters, then the bass with 2, the lamprey with 1, followed by the outgroup with none. Knowing how phylogenetic trees are built should help in understanding how to read and analyze them. More recently, scientists have been using protein sequence evidence and DNA sequence evidence to build even more accurate phylogenetic trees, rather than simply relying on morphological evidence. The reason why this is the case is because morphological evidence may lead us to believe that species are more closely related to one another than they in fact are. An example of this could be that both penguins and whales have a layer of fat beneath the skin called blubber, useful for insulation in cold environments. But penguins are birds and whales are mammals. They belong to 2 different lineages of animals with an extremely distant common ancestor, well over 300 million years ago. If we were to rely on a piece of morphological evidence like blubber, that might suggest that they are more closely related than they actually are. DNA and protein sequence evidence is helping to clarify more accurately the relationships between species. Another piece of evidence that we can use to build phylogenetic trees are molecular clocks. When building trees, knowing exactly how far apart 2 groups of organisms are is a critical issue. Remember, phylogenetic trees are used to represent how much time or genetic change has occurred. So estimating when a common ancestor existed for a group of its descendants allows for a more accurately proportional model to be constructed. What molecular clocks rely on is the accumulation of mutations. Mutations occur at a relatively consistent rate between groups. So if we can calculate how many mutations have occurred in 2 different species, we can make an estimation as to how long ago those currently living species diverged from their common ancestor. In this chart, each of the dots represents a currently living species. It shows how many mutations have accumulated in those species relative to one another and a common ancestor. This point right here represents a species that has accumulated more mutations than any other of the species. It diverged from their common ancestor around 120 million years ago. As a side note, molecular clocks require a really complex set of calculations to calibrate them, and that's why we don't rely on just a few data points like the 3 green outliers, which have clearly shown, for whatever reason, a much slower rate of evolution. Using multiple data points allows us to establish a more accurate trend. We can use a chart like this to estimate how long ago an organism diverged from a common ancestor. For example, if a species is found to have accumulated 60 mutations, then we could estimate that approximately 95 million years have passed since it diverged from its common ancestor with the other species. It is important to note that phylogenetics is an ever-changing field. It relies on model building, and as we accumulate new information, as we gather new evidence, as technology improves, and we have more refined techniques for establishing links between organisms, phylogenetic trees are always being tinkered with. As our understanding grows, the relationships between organisms are revised. Sometimes moving organisms farther apart from one another as we learn new evidence that separates them, or moving organisms together as we learn new evidence that makes them more similarly related. That wraps up these 3 topics. Thank you for watching. Until next time, take care.